Welcome to this afternoon session. I am Honghui Zhen. I'm an investigator of cell and circuit genetics at the Allen Institute. I'll be your MC this afternoon. First, I'll introduce the third team talk. This talk will be about building single neuron models from mouse cortical neurons using our in vitro single cell characterization pipeline data. And presenters will talk about how the modeling needs actually influence our choice of different stimulation paradigms for our electrophysiological recording, single cell patch recordings. And they will describe the models that they generated, different kinds of models they generated, and how they perform for different kinds of stimuli. The presenters are, who will be first? Corinne Teeter, Nathan Gowens, Jim Berg, and David Pham. Welcome.
All right, so today we're going to be talking about the work that went into putting together the Institute's first pipeline focused on the generation of computational models. And so as Honghui mentioned, this basically involves taking the data that's present in the Allen cell types database, using that to build models of individual neurons, and then presenting those results alongside the original data in a publicly available format. So this work is related to two of our broader institute goals. So the first one is to define and characterize the cell types of the cortex. And a lot of the ways that we're doing this are experimentally based. So in terms of measuring electrophysiological features of the firing patterns of different cells, characterizing their morphologies, and doing transcriptomic profiling on single cells. And you've heard about some of this mentioned all throughout the day. Another goal is to simulate cortical functions that underlie visual processing. And to do this, we're building models that consist of networks of neurons, and then we present visual stimuli to those models and analyze their responses. And so the single cell modeling work really links these two goals together and tries to bridge kind of our components focused work on characterizing the cell types to our computation and even cognition focused work on the modeling side. And it does that by allowing us to populate these network models with the single cell models that were based on cells that we've experimentally characterized directly. So you might be wondering why we're building models at all. And so at a really high level, models are things that attempt to explain specific experimental observations by evaluating the fundamental principles involved. So building the models helps us figure out and hone in on what's really important for a particular phenomenon. Models also generate new experimental questions. They provide guidance for additional measurements. And this is really important at a place like the Allen Institute, where the experimentalists and modelers can work so closely together. And finally, models represent biological concepts in a quantitative form, but it's simplified to, one, allow us to actually do things with the models and predict and make predictions with those models. But also, simplification helps us actually clarify our understanding of what's actually going on. So, sometimes when you have so much data you don't know what to do with, you need to simplify it to get a better intuitive sense of what's happening. A lot of times we'll just talk about kind of making a model, and it's not really clear the steps that go into that. So again, I'm just going to talk at a really high level about how you make kind of any model. And there's basically 4 steps you do in kind of regardless of the level of complexity of the model that you're trying to develop and evaluate. So the first thing you do is you start out with some data that you're trying to explain. And so in this case, we have just a set of data that relates 2 variables together. And we have an idea about how these two might be related, and we can express that in an equation. This is the most complex equation you're going to see in the presentation, so don't freak out. Basically, we have the idea that the variable on the y-axis is just something about the variable on the x-axis multiplied by some constant. So to evaluate whether or not this is a good model, you can basically split the data in half and then use half of the data to test different parameters. And this is called optimization. So you're trying to hone in on the parameters that best describe this particular data set. And so in this case, it looks like A2 is the best fit. And we call this process training the model. Now, we're not done. We need to evaluate how well this model performs on new data, so we can use the other half of the data that we left out and see how well our line describes that data. And in this case, it's doing pretty well, and we call that process testing the data. So this is overall, again, the things we're going to be talking about will obviously have a lot more complexity than just fitting a simple equation, but they still boil down to these 4 essential steps. Okay.
So we created this modeling pipeline to address 3 research questions. The first is, how does the level of abstraction that we use influence our ability to actually predict the firing patterns of neurons? The second is, how much do the parameters that are fit by neurons actually inform cell type classification or characterization? And the third is, we would like to analyze the network behavior when we use our specific cell types in our network models. So in order to address these questions, we basically have made models on many different levels of abstraction. These include biophysically detailed models, which have very specific representations of ion channels, and they have many parameters. They include using the reconstructions of the somatic and dendritic morphologies, and they can predict the membrane potential throughout the entire cell. We also use or create very simple neurons where the entire neuron is modeled as a simple spherical ball. And basically the point of these, the biophysical models can aim to actually produce the spiking behavior of the spike itself. However, in glyph neurons, we aim only to produce the spike times represented in this roster. And there's basically benefits and drawbacks to using any types of models. So the biophysically detailed models are much more computationally intensive. They have more parameters. But the benefit is that these parameters are linked directly to biological mechanisms, where that's not necessarily the case with glyph neurons. However, glyph neurons are much less computationally intensive. And their scientific benefit is that because they're simple, any increase in model behavior you can directly corresponds to a very specific phenomena that you've added to the model. So both of these types of models are used in the field extensively. And I'll start by talking a little bit about our generalized leaky, integrate, and fire models. And with these models, we basically utilize the simplicity to ask how, when we add biologically inspired phenomenological rules, how does that improve the firing behavior of our models? So basically the starting point for all glyph models are the classic leaky, integrate, and fire model. And in this model, the complexity of the membrane is reduced to a simple RC circuit, and we inject current into the circuit. And here you see a schematic of how a leaf neuron would correspond to current injection. So if you inject current here, what you see is the voltage of the membrane increases until it hits a threshold. Now this threshold would correspond to the same sort of threshold that you would have in a biological neuron. And at that point, a spike happens, and the voltage is reset to rest. And then again, the voltage can go ahead and charge until it spikes again. So now we're going to add phenomenological complexity that corresponds to directly to things that we see in biology. And although I'm showing you schematics of this behavior, all the rules that we're showing here are fit directly to the IVSCC data. So first, we know that real neurons, they don't actually reset to rest. So I'm showing this here. When the neuron spikes, it resets, but it resets to a voltage that's not rest. It's something that we've measured from the data. And correspondingly, ion channels can become inactivated. We can model that by an increase in the threshold when the neuron spikes or the model spikes. And again, we fit this directly from the data. Also, we know that ion channels, ion currents can have long-term effects on the spiking behavior of the cell. So when a neuron spikes, we can go ahead and inject another current that represents the current coming in from the ion channels very abstractly. Again, we fit this data specifically from the IVSCC data. And then we fine-tune the amplitudes via our optimization process. We also know in real neurons that the threshold can adapt due to inactivation or activation of voltage-dependent channels. So you can see here that the threshold is actually fluctuating based on the voltage of the neuron. And finally, we know that due to these voltage-dependent ion channels, that the subthreshold behavior is not actually perfectly linear. And you can see that by when you inject a ramp into the neuron, instead of seeing a linear voltage response, you actually see a curve here. So we basically incorporate all of these different combinations of these phenomena, and for any of the results or the specifics of the optimization process, go ahead and stop by my poster this afternoon. And now Nathan is going to talk about biophysical models and show a really nice example of an optimization process. And although the specifics of the optimization are different for glyph neurons, the basic idea is still the same.
Yeah, so Corinne just described the kind of variety of glyph models that we've produced, and so now I'll walk through how we put together the biophysically detailed models. So again, one of the differences between the biophysically detailed models and the generalized leaky integrate and fire neurons is that in the biophysically detailed case, we're using the reconstructed somatic and dendritic morphology of the original neuron. So we can basically model the potential across the entire cell. Then we take that morphology and we place active conductances at the soma. And these active conductances are really meant to more directly tie to the actual ion channels that are found in the particular neuron. So they're meant to behave as those ion channels behave and also represent the diversity of ion channels that might actually exist in a particular neuron. So there's a more direct tie to the actual kind of biological, ion channel species. The next thing we do is we look at the responses of that neuron to a step current injection and measure different electrophysiological features from that original neuron. So things like the spike height, the spike width, how fast the cell is firing, a variety of different features. And then we can look at our model, which has just kind of random densities of active conductances here, and then try to keep adjusting those conductances until the model features match the experimental features. So the model is in red, the experiment is in black. You can see that kind of starting out with just a random set of conductances, we're not at all close to what the original cell looks like, but through an optimization procedure that Corinne just alluded to, we can basically iterate toward a solution that looks much more like the original recording. All right, so you can see now the overall firing pattern is much more similar and the overall spike shape is much more similar to the original cell. So now that we have this model, which has been trained on this particular type of data, we can take it and apply all kinds of different stimuli and compare those to the original experiment where we also have a variety of stimuli. So for example, with the biophysically detailed models, they were trained on these step currents, but we can apply a noisy current injection to those and see the kind of patterns of spikes we get. If you look between the experiment and model, you can see the overall pattern is relatively similar between the two. Maybe the model is firing a bit faster than the original experiment. We can apply slow ramp current injection, and you can see that the model and the experiment are starting to fire at similar times. Maybe the spikes here are a little bit shorter than the original experiment. And we can do a similar thing with the glyphs. Again, with the glyphs, we're basically looking at spike times. So those have been trained on noisy stimuli, so the kind you see here. So we can test those with these step current stimuli. And you can see that kind of the overall firing rate is similar between the experiment and the model. Maybe the model is firing a little bit slower at some of the lower stimulus intensities. And similar things you can do with the ramp. You can see the firing kind of rate of adaptation is similar, but the model is starting to fire a little bit later than the experiment. So we've developed these techniques for creating models based on data from individual cells that we can link to all the other data we've collected about those particular cells, their location in cortex, Cree identity and things like that. And so we have these techniques set up, and now we need to apply those in a pipeline type way.
Okay, so now we know why we need models, and we know what the models need in terms of data, and so that's where I come in. I lead a group in structured science to generate patch clamp EPHIS data as part of the in vitro single cell characterization pipeline. And so what we thought we'd do is run through kind of the lead up to the May 2015 data release and kind of go through some of the processes as to how we got those models on the web. So to go back to the beginning part of the project, way back in May of 2014, a year prior to the data release, we pretty much had one patch clamper in my group patching cells. We were kind of drying out from the flood that had just been in the CVB, and we were generating little bits of preliminary data to get feasibility studies to say we could actually generate these models on site. And so this is just a slide from a Mindscope Advisory Council where we're showing that preliminary data. And what we had is modelers a lot of times were in the room with us. We were transferring data on thumb drives. A lot of emails were going back and forth. And it was a lot of fun, but it wasn't tenable for our eventual goal, which is basically to have a large group of patch clampers as the team grew, and a large number of modelers, both in-house and producing data from modelers outside of the Institute that they could access and use. And so as we started to move towards this, we realized there was a lot of challenges. We had to design a stimulus set that was going to be applicable to the most numbers of neurons. We had to bake in some flexibility. And then with the sheer amount of data that we were going to be producing, we wanted to minimize how much handling we had to do of it. And then we had to watch out for these rare cases, cells that were going to break the system. So to deal with all these challenges, we relied on a third member of this team, the technology team. So basically, it's kind of three different groups from different parts of the Institute. We really worked together on kind of a really day-by-day, sometimes hour-by-hour basis in the day to really kind of come together to get this product on the web. And what I hope to impress upon you is that it wasn't one or two big things that we did to kind of do that, but it was hundreds and hundreds of really, really small, almost trivial modifications on each one, looking at it on its own, to kind of combine to get the data out. So I'm going to highlight some of these small things that we did to kind of move the data forward. Interacting with the modelers, you've heard from the intro that the biophysically detailed models and the glyph models require very different stimuli. So it was kind of my team's job to their job was to tell us what they needed. Our job was to give it to them without killing all of our cells. So what we came up with was collaborated with the biophysical models to give these long 2-second, we call square stimuli that give them lots of action potentials to be able to fit each of the shapes of the action potentials in the train. Whereas the glyph models, we collaborated to generate a noisy stimulus that simulates, that's reflective of the natural voltage variation that we see in vivo to give them lots of spikes to look at that spike time. And so it was very different, but one thing that was in common is that we repeated all of these stimuli multiple times, 3 or 4 times, so that they could have that test data set that they could leave out at the end. And so as we got into production and started producing more and more data, what we realized is that the modeling field starts to move, and the pipeline was set. And so what we had to do is kind of bake in some flexibility to deal with that. The influence of as they're getting data and shifting their models. So what we did is we basically kept some of the stimuli exactly the same that we'll probably give to every single cell that we ever record from here, whereas we allowed some flexibility to kind of swap in some different stimuli. And so I'm just showing a very kind of brief example of that in this noisy ramp to Rio base that's required for some of the more complex glyph models, where initially it was spiking too much and they weren't able to fit the pre-spike nonlinearities well enough, and so we modified it to basically give a more shallow ramp, and that they were able to use to get the accurate models that eventually went on the web. And so it's really nice to have this flexibility, but it brought up some issues, and one of the issues is just kind of communication. How do we communicate when we have hundreds and hundreds of cells? Which cells have this stimulus versus that stimulus? And so that's where we really relied on the technology team to kind of guide us. And so this may seem like a really, really trivial thing, but before we moved into production, we had some random stimulus names that really made a lot of sense to us, but were difficult to kind of communicate. So we standardized to this 16-character name, and we produced a glossary. And so in the previous example, when we would switch to a different stimulus, we would update the glossary, and this saved about 1,000 emails, I think. It was really incredible. The other thing that we did that was probably more important is that we really standardized the shapes of the stimuli. Where before, as we were going, we were kind of flying by the seat of our pants, and we'd stimulate different neurons different ways. Whereas what we did, kind of giving a specific example, is we standardized a 1-second rest period with a little, we call a test pulse at the very beginning of every single data trace, so that no matter what the data trace that was coming in, the technology team would know to look at that first 1 second, and it would always be identical, so we could compare between sweeps. And so that led to basically, previously our quality control was that we would give a qualitative ranking of 1 to 10 in terms of good recordings and bad recordings. But what we were able to do is take that 1 second of data and analyze it for membrane stableness and how things might be changing throughout the experiment, and also automate a lot of that QC. So it just, the data was able to be seamlessly transferred to the technology team, which David will take from here.
So Jim just talked about some of the standardization and automated processing that we did to hopefully standardize the product a little bit. And so now what I'm showing you is the actual IVSCC data processing pipeline. This is a workflow chart. All right, so every little box here is a module that does some processing. It uploads the results of the processing to a backend database, our LIMS, our Lab Information Management System, and then hands off to the next module. And this all happens in a mostly automated fashion. So it took a lot of meetings with Jim and everybody, all the modelers, to decide exactly how we wanted to represent all this information in the database, which modules are important, and what order they need to happen in. And so you have to ask yourself, why is it worth it to go through all this hassle to design this pipeline? It was a lot of work, and the answer was yes. And the answer is yes because there's a lot of data coming through, and you don't want to have to manage all of these things yourself anymore. So other benefit is that the data is now backed up and stored systematically in a database, so now everybody can query for particular cells they're interested in. Other big benefit is the technology team has dedicated QA resources, so there's people that have their eyes on all these modules at all the time, and if something goes wrong, they'll notify the important people. And finally, technology uses the database representation of all of this analysis and all of this data to build the product. So I just want to talk about some of the interesting things we learned when we designed this data processing pipeline. The IVSCC pipeline was a new experience for, I think, both the technology team and also the modelers, because this was the first time we actually tried to integrate research code into our pipeline. This was not code that we necessarily wrote ourselves. And this was, what do I mean by research code? So research code, I think people who are doing analysis in maybe a more typical academic setting are used to doing bulk of the data collection first and then having a stable data set that they can fine-tune their analysis to, right? And now a pipeline is slightly different in that you have data that's continually trickling through at a more steady rate, and that data maybe isn't stable, right? The stimuli are changing slightly, and your code needs to be robust. It needs to be robust enough to maybe handle these slight variations, but also it needs to be robust enough to fail in an expected way. So if something goes wrong, errors need to be reported in a way that we can forward those errors to the right people. Now, the IVSCC pipeline was particularly interesting because we added the extra layer of complexity that we wanted to publish some of the code we used to do this analysis so that people could reproduce the results that we generated onto our website. And this imposed a pretty serious set of restrictions. The code now needed to be clean, it needed to be documented, it needed to be well commented, and it needed to be easy to use, not just by us. So given all of these constraints, you have to ask yourself, like, well, is it actually worth it to integrate code into the pipeline? And in fact, for the biophysically detailed models, the answer was no. The answer was no because Nathan's optimization code was, it was evolving at a pretty rapid rate so that he could produce high quality models that we could upload at the end of the day. And so we let him simply produce results and upload them to the database as opposed to running them in an automated fashion. So just very quickly, some of the lessons we learned along the way for generating this pipeline. Number one, super important to start collaborating early, especially in terms of code design, just to make sure that there's no traumatic failures that are going to happen later. But second, it's also really important to integrate that code late once the code is stable and mature, because the pipeline, as I've shown in my image here, is heavy and moves slowly, and it's hard to change. So, but it will continue to move regardless of what you want to have. Anyway, and then finally, super important also is to finish early because that leaves more time to do fun visualizations and product design. So now very briefly, I'll just describe to you at a high level what we do when we design the product. First and foremost, we're trying to answer the question, how do you want the public to interact with your data? This is a lot of data that we've just generated, and it's going to be available not just to your scientific peers, but also to the general public. And we do that by trying to create example questions that our users are going to have. We call these use cases. And then we try to optimize the experience on the web page to make it easy to answer those questions for everybody. And sometimes that means computing additional features of the data and building custom navigation visualization tools. The last thing I'll say about this is that the product is actually a really unique place where all of the work of a huge number of people is now being represented in one place. All of the data is there, all the annotation is there, all the analysis is there, and now we've built custom navigation tools to make it really easy to browse and find problems. So every release, last May of 2015, for example, we have a bug bash. That bug bash is there to help us find problems in the web application, but it's also there to help us find problems in the data itself. And we did find problems. Now, very quickly, I'm just going to show you how to find the models in the web page, because they can be a little bit tricky to find. So we've seen this slide a few times now. This is what happens when you go to celltypes.brain-map.org. All these dots are different cells. We can filter that list to just the cells that have glyph and biophysical models. If you click on the ePhys thumbnail on the right for a particular cell, it'll take you to a page that shows you all of the recordings of responses to stimuli for one cell. You can pick a model and do a side-by-side comparison of the cell's response and the model's response to the same stimuli, which is cool. And now just digging in a little bit for one of the biophysical models, you see that the model at low short square stimulus amplitude is a little bit more subdued than the actual cell, but then as you increase the amplitude, it looks a little bit more similar. The last thing I'll say is that, as I said before, we publish some code to help people reproduce the results that we've generated on our web page. All of that's on GitHub right now. We call this the Allen Software Development Kit, this Allen SDK. For the cell types database, this involves a lot of code for downloading cells in a regular file format we're calling Neurodata Without Borders. And also the models themselves. People can then download the models, run the models on custom stimuli or the stimuli we presented, and see what they do, which is pretty cool.
All right, so now we have this database of models alongside our experimental data, and we can start to use those models to address some of the questions that we brought up at the beginning of the presentation. So we can do things like look at how the level of abstraction affects our predictions of a cell's activity, because we can make both biophysically detailed models and glyph models from the exact same neuron, and we can do that for many neurons and systematically go through and compare and contrast the different types of models. We can also use the model parameters of all the models in our database to see if they can complement our cell type characterization. So you can do things like what's shown on the left, which is basically plotting different model parameters against each other, in this case from the glyphs, and see what kind of different clusters or different areas of space that those models represent, and then see if those connect to different cell types. On the right, you have the set of biophysically detailed models, parameters, and you can compare those to other things we know about the cell, like the Cree identity of those particular cells. We can also use these models as the building blocks for our network models. So on the left is a biophysically detailed model of layer 4 that's been populated with actual models from the cell types database, and you can see here the response to showing that model a visual grading. And then on the right is showing a glyph model of cortical column computations, and we're planning to, again, populate those with the individual glyph models that represent the different cell types that are present in this model. So just to wrap up, what we've done and described is the development of a standardized modeling resource that sits alongside the original data, which is available to the broader neuroscience community. We've used these models to characterize the fundamental components of the cortical circuitry. And those components form the building blocks for future investigations of cortical computations, both here at the Institute and for the broader neuroscience community. Finally, doing this modeling work draws on a vast amount of data that's produced by the Institute. So we just need to and want to think, probably a third to half of the people, shown in this picture contributed in some way to the results that we've been talking about. So thanks to all of you. Thanks to Paul Allen for his vision and support. And thank you all for your attention.
So when you adjust the biophysical parameters, the conductances of the different ion channels, do you also use gene expression data to see what channels are actually expressed in those cell types?
Yeah, we have not at this point. I think that's something we definitely would be interested in doing. Part of that is because our conductances are somewhat genericized to try to represent kind of the full range of different variations of conductances that are actually in the cells. But that's definitely would be an interesting direction to look at, yeah.
I see that you look at layer 4. Are you going to look at any other cell types in other layers?
No, sorry. So the model I showed at the end is just a layer 4 model, but that's because we're starting at the input stage and we'll continue to build complexity. So yeah, just the current state.
From the experimental data set, I'm curious, if you were to define cell types purely based on the electrophysiological, the diversity of electrophysiological responses, how much does that agree with taking the molecular approach and defining cell type diversity from that angle?
Very good question. We're just getting into that, and we're at it. We're actually kind of combining the transcriptional approach with the electrophysiology approach right now to answer that exact question. So I don't have an answer right yet, but hopefully soon.
Do you know how people in the outside community are using these data? I mean, I understand how you're using them, but how many people have gone to the site? How many people have played with the data? Have you gotten any feedback?
I don't have like web statistics off the top of my head, but I will say that we're, sorry?
It's like, I think 3 million page views of the cell types.
That's a lot. I don't know the answer to that question.
Dwell time?
I do that regularly. Sorry. I will say that we're actively pursuing collaborations, especially with other large-scale modeling initiatives like the ModelDB and the Blue Brain project, and we know that people are going to be using these models, and we're hoping to integrate with them pretty actively.
Okay, let's thank the multidisciplinary team again.